All right, let's use the information we've learned about enzymes and apply it to a different enzyme, one called catalase. So let's read a little bit about the background information in this lab and see what happens when catalase is broken down within our bodies. So the enzyme lab, hydrogen peroxide, and you may be familiar with this chemical. It's found in a dark bottle, and your parents may have put it on a cut or a scrape when you were a kid, or even now. And when you do that, it produces bubbles. Okay, well, naturally in our bodies, hydrogen peroxide is a harmful chemical produced during metabolism. Your body is actually producing hydrogen peroxide within your cells right now. Some organisms have higher metabolism than others, and therefore these organisms are going to produce more hydrogen peroxide. Well, to prevent damage to your cells, hydrogen peroxide must quickly be broken down, and it's broken down into less dangerous byproducts. And those byproducts are water and oxygen, things that are normally found within our cells. Your body uses an enzyme called catalase to break down hydrogen peroxide your body produces. Organisms with a lower metabolism need less catalase than those with a higher metabolism. During this lab, you're going to be asked to determine how much catalase enzyme is present in certain types of cells. You'll be measuring how much hydrogen peroxide is broken down by different types of cells. And you can determine this by reading the amount of bubbles produced when exposed to the catalase enzyme. So take a look at these pre-lab questions and fill in your answers. All right, after you finish your pre-lab questions, we're going to take a look at what this process looks like using a little diagram to help us understand what is the enzyme, what is the substrate, and what are the products. All right, for this lab, it's important for us to identify what's our enzyme and what's our substrate. Remembering that this is our enzyme as it remains unchanged, okay, from B to C to D, and this is our substrate, okay, as it gets broken up into products throughout the reaction. All right, so our substrate or our reactants in this lab is H2O2. Okay, hydrogen peroxide. And once again, like mentioned in the background, this is a byproduct of metabolism and is present in the cells in our body right now. For our lab, we're going to be adding the hydrogen peroxide into or onto the cells to see the amount of catalase present. So B, our enzyme, is catalase. Okay, oftentimes, oops, I forgot an A there. Oftentimes, enzymes end in ASE, and that's helpful in being able to recognize what is the substrate and what is the enzyme. All right, so catalase, once again, breaks hydrogen peroxide down into less harmful byproducts. In this particular lab, H2O2, hydrogen peroxide, gets broken down into water and also oxygen. Oxygen, we saw in the lab, as a byproduct as bubbles. We measured the amount of bubbles. Bubbles. Okay, once again, right here on our enzyme is our active site, and that is where our hydrogen peroxide fits directly into that catalase enzyme. Remember, enzymes are like locks and keys. They are very specific as to which substrate they act upon. Ms. Minnick is going to show us what this looks like at a molecular level, which molecules are going in and which ones are coming out. Now we're going to go to Ms. Minnick and look what this looks like at a molecular level. All right, now you're going to take your data that should look somewhat like the data presented, and you are going to answer the conclusion questions, thinking about what is an enzyme, what is the substrate in this particular lab. And Mr. Fuller is going to help us think about enzymes and substrates and wrap up this lab.
Now that you've completed your experiment, you should have collected some data that can allow you to make some interpretations about what's going on in this activity. Hopefully what you saw was lots of bubbles in the muscle and liver cells in the raw and unaltered foods. There was a few in the carrot and potatoes, and the starches and the plants. And then once you put these foods into either a cooked water bath where you would have heated them up, or changed their pH by putting them in vinegar, that there was a drastic change in the amount of bubbles that occurred when you added the hydrogen peroxide. Now what we're going to do is take a look at why that might have happened. When we talk about enzymes, and in this case the enzyme we're talking about is catalase, they're very specific to what they bind to. And as we've learned, that the shape of the protein is extremely important in the function of that protein. So if anything happens to that shape, it's not going to function very well. So looking at the example up here, if you look at letter A, this would be our substrate. And the blue colored object that's labeled letter B would be our enzyme. As you can see, under normal working conditions, as A combines with B, all right, so the substrate combines with the enzyme and goes through its process, the substrate gets broken down into two separate units. So the enzyme has helped in that process. It has helped act as a catalyst. It has helped break that substrate down. And if you look even more closely, there are specific shapes that are on both of these objects. Now, as long as these shapes stay the same, it's going to work. But let's say, for example, that this right here were to turn into a triangle shape. Now, most of you probably would be able to see that this square figure right here, there's no way that that's going to fit. This will not work. Okay, that's not going to fit. Therefore, this reaction will not take place. Now, what we call this, when these shapes or the enzyme changes or it gets compromised, it's called denaturing. All right, or to denature something. And when an enzyme gets denatured, the shape changes, it loses its 3D shape, and therefore it loses its function. So, when looking at this, when we put the food into the hot water, it became denatured. When we put the food into the vinegar, it became denatured. And that's because things like heat, changes in pH, all right, so the vinegar was acidic, we changed the pH, or even changing something like the salt content can really have an impact on these enzymes because they're extremely sensitive.